Hello everybody, we are talking today about local DNA statistic. What we mean by this? Well, first remember that the statistic is about making sense of data, of extracting information from data. In genomics and molecular biology, we are looking for biological sense. The story that we want to tell, that we can tell from the data, can be global, meaning about the complete genome, or it can be local, about some part of the genome. In the last class, we talked about global properties like GC content and GCSQ. Now, for local statistics, we are going to introduce a new concept. We are going to speak about a window that represents a part of the genome from an initial position and with a fixed size. For example, we may like to analyze a region of 500 base pairs starting at the position 3000 in the genome. So let's start with the basic thing. We will load the E.coli genome with the Sequiner library. We use read FASTA or the correct file. We check that we have only one chromosome, and then we check that chromosome has a length of 4.6 million base pairs, which correspond to the length of this strain of E. coli. To calculate the GC content, we will recycle the function that we used last time. We just shortened the name. This is version 3, but we don't need to write it. This is the only version we are going to use here. It takes a DNA sequence, get it to uppercase, counts the number of C and the number of G, and then essentially give you C plus G divided by the total length of the fragment. And we test it by applying to the complete genome, and it makes sense. Perfect. Now, what we really want to do now is to calculate the GC content window by window. Therefore, the result should depend on the position of this window, the size of the window, and of course the genome or the sequence that we are carrying. So let's solve it in plain English first. The inputs that we need are the position, size, and DNA. These are given to us. Someone will take care of that. We don't care right now. We are writing the function. The user will tell we are writing the function. We know how to calculate the GC content of a complete sequence. We have a function that we call GC content. We have this function. We don't need to make it again. We only need to cut DNA in a fragment that's starting at position and from the length of size. And for doing this, we will need index. If you remember from the previous course, to look inside a vector, DNA is a vector of character, we need a set of indices starting at the position and with the corresponding size. So the first version of this program is, or this function is simply this. You write the name of the function, you have to find a good name, you write the keyword function, then the inputs, in this case position, size, DNA, the English description of what we want to do. This hash mark means comment. It means that everything that we put after that will be a comment and will not be executed by the computer. It's just a message for us. This is for humans. And at the end we will return some answer that we have to calculate. Okay, for solving this part, we should remember something from the previous course, from the first class of the previous semester. The sequence function, that is not a DNA sequence, is instead a sequence of number. It has several inputs. All they are optional, but you have to choose them carefully. You can specify from value to value, and we'll fill them, or we can say from and how many we want. And the two will be calculated automatically. And by will allow us to go 10 by 10, 20 by 20, or whatever we want to specify. It will specify the step. So, for example, we have the sequence from 5 to 14, or we can also say a sequence from 5 with 10 elements will be the same 5 to 40, or in some cases we will like to specify an initial value, a final value, and then how much we should increment every time. The step size is 3, and you go from 5 to 8 to 11, 14, and so on. So this will help us to, for example, find what is inside the genome. We can use the sequence of number from 300 and length 500 as an index of the complete genome and assign this to a window. So this function gives us a vector of numbers, and inside the bracket we use it to get one part of the genome of size 500 starting at position 3000. In general, we will start at start and we have a size of size. These are parameters that we can decide later. So let's finish our code. We have the function name and we have the explanation, and now we fill the missing part. The indices will be chosen using the sequence function from position, initial position, and with the length of size. Then we use these indices to cut DNA to extract only the relevant part. We call it window. And finally, we use the function gene GC content that we had previously to calculate the GC content only of this window, and we return this value. If you are clever, you can write this in one line. To do the next step, we need to move through the genome in several windows of a fixed size. So you have the complete genome, and we will go step by step with windows that are relatively long, let's say 1,000, 10,000, 100,000 nucleotides, and we take a value, and then we move to the next one without overlap. So let's say from 1 to 1,000, then from 1,001 to 2,000, 2,001 to 3,000, and so on. These are called window, more technically sliding windows, in a way to see all the genome piece by piece. So we need to create a vector with the positions. We know the length, but we know it needs the positions. We will store in a vector. As usual, we create a vector using sequence. We start from the first position in the genome and until the end. Now there is a detail here. If we are not careful, we will step out of the genome. If we know the size of the window, this is the size of the window, we have to take care that we don't fall out of the genome. And that gives us a vector. And now we do the magic. We have the positions and we have our function, and we just apply window GC content to each one of the positions, and we add extra parameters here. So let me go back 1 second. I want to show you something. Our function takes 3 inputs. This is different from before. We have 3 inputs: position, size, and DNA. When we use the function in supply, the first parameter will be taken from the input vector. Then we have the function that we are using. But if we have extra parameters, we put them at the end. So it will take one by one each one of the position in WinPos, and we will call window GC content with extra parameter window size and genome. This changes every step. These don't change. They are fixed through all the calls to this function. We do so. We create, for example, window position from 1 to length genome minus 10,000, and we are going a step of 10,000, and then we calculate the GC content in a vector, applying the function window GC content over the vector WinPos with a size of 10,000 over all the genome. And then we plot this vector, and you get this nice drawing here. Some of you may remember from the previous course that we can use the pipe symbol to connect successive applications, and the result of the first is taken as the first input of the following, and the previous expression can be written here in one long line. We create the sequence, Then we apply the function to every element of the sequence, and then we plot the resulting vector. Now, let's think about the second statistic that we use, that we learn about DNA. Let's talk about the second statistic that we learn about genome, the GCSQ. We want to calculate this ratio using several windows and see what happens. We again recycle the code that we have from the previous class. We test with the full genome, nothing surprising, everything works fine. Then we write a new function called window GCSQ, following the same shape that we have for the window GC content. The same logic, the only change is the function that we are using inside. Then we apply exactly as before, create a vector with all the starting position. Then we apply the function window GCSQ over all the windows, and then we plot. In this case, I'm using some extra nice drawings, and I put an horizontal line just to get an idea where is the zero value. And we see this interesting pattern that from one part of the genome, the sign of the GCSQ tends to be positive. Of course, you have some exception, and then it tends to be negative with some exceptions, and then positive again. So we have a mostly positive side, mostly negative side, mostly positive side. Why? Well, as we discussed previously, this happened because of the way that bacterial DNA is replicated. The GCSQ change the sign at the boundaries of the two replicors, the part that are used for replicating the chromosome. This is the biological part. You have the origin of replication, where the DNA is split in two halves, and replication happens simultaneously in two replicors, the left and right replicor. And in each replicor you have this phenomena that the polymerase is filling immediately one of the strands, and the other is filled in a slower rate, and that exposes part of DNA to the environment, and that eventually can trigger some mutation from C to G. This is something that happens in all bacteria and all organisms that have only one replication origin. Archaeas and Eukarya don't have.